Welcome. In this presentation, I will walk you through the paper with the title Versatile Emulation of Spiking Neural Networks on an Accelerated Neuromorphic Substrate. My name is Sebastian Biodel and I am a PhD student at the Kirchhoff Institute for Physics at Heidelberg University in Germany. I am only one of the many authors who contributed to the content of this paper, and this very extensive author list on the one hand tries to acknowledge those who have played important roles in building this complex system, but it also reflects the fact that this publication displays 5 different, distinct experiments to emphasize the versatility of our neuromorphic system. Wherever available, I refer to further publications, and in the upper right corner you will find QR codes. If you download the slides, these will also function as links. The neuromorphic substrate mentioned in the title is Brainscales 2. It is a family of mixed-signal ASICs which are fabricated in a 65 nanometer CMOS process. Just as the first Brainscale system, it emulates spiking neurons with an acceleration compared to biological timescales. For Brainscales 2, we specified a speedup of 1000. That means that 1 second in the biological time domain is emulated in only 1 millisecond of wall clock time. The analog core is the heart of this system. It features 512 neuron circuits, which implement the adaptive exponential integrate and fire model. They can also be configured to exhibit pure LIF dynamics, and each of their parameters can be individually controlled using biases and references generated by an on-chip parameter storage. You can connect multiple neuron circuits together to construct larger neurons with an increased fan-in, or to form structured neurons with multiple compartments which are then connected via intercompartmental conductances. Each atomic neuron circuit receives input via 256 synapses. Each of these synapses has a 6-bit weight. The most notable addition compared to Brainscales 1 are the digital plasticity processing units. These embedded processors are freely programmable and interface the neuromorphic core via SIMD vector units. We call this concept hybrid plasticity. This scheme couples a flexible digital processor with a multitude of analog observables. For example, local sensors within the synapse circuits measure the correlation between pre- and postsynaptic spike trains. This, for example, allows to implement STDP-based learning rules. Plasticity programs can further incorporate the neuronal firing rates and even the neuron's membrane potentials. To digitize such analog observables in an efficient manner, parallel ADCs with a total of 1024 channels are tightly coupled to the plasticity processor's vector units. The PPU has also access to the on-chip configuration bus to read and reconfigure almost all subsystems of the ASIC. On the one hand, these processors can be used to implement a wide range of plasticity rules, but even further, they can be used to set up or even calibrate the system. Especially, they can be programmed to simulate the environment for closed-loop experiments directly on the ASIC itself. In a bit, I will introduce you to 2 experiments that make use of this. The architecture of Brainscales 2 allows to implement many different experiments. In this paper, we have collected 5, which feature different aspects of the system. Let us start with an experiment on sampling-based Bayesian computation. Here we observe a recurrent network of LIF neurons, shown on the left, and in a previous publication it was shown that these networks can approximate Boltzmann machines, where we define a random variable over a collection of such binary neurons based on their refractoriness. A neuron which is in its refractory state is assigned the value of 1, and otherwise it represents a 0. On the right hand side, we train such a network of LIF neurons on Brainscales 2 to represent an arbitrarily chosen probability distribution over 5 neurons. This target is shown in blue in the bottom plot. The histogram shows how the sample distribution approaches the target distribution during the course of the training. In the upper plot you see the evolution of the Kullback-Leibler divergence, which is a measure of the difference between the sampled and the target distribution. With such networks, you can implement discriminative as well as generative models, which you can use for inference and completion of, for example, vision datasets. For further information, please refer to the publications highlighted above, which focus on related topics investigated in simulation or the predecessor system Brainscales 1. The second experiment we want to discuss demonstrates a reinforcement learning task on Brainscales 2. It was implemented primarily by Timo Wunderlich. On a smaller prototype system, a network of 32 LIF neurons was trained to play a simplified version of Pong. The analog neurons encoded the state of the network as it is shown on the left. The most active neuron determined the position of the paddle. Similarly, the current position of the ball was encoded by the input population. The network was then trained using a reward-modulated STDP rule, utilizing the success of the network in playing Pong as a modulatory signal. The corresponding publication highlighted two main results. First, a suitable learning rule can intrinsically compensate fixed pattern noise present in such an analog substrate, and this in the end reduces the requirements on calibration routines. In my opinion, even more interesting is the fact that this experiment was the first to demonstrate a closed-loop setup fully contained on such an ASIC. The game environment was simulated on the PPU, the host system and the FPGA were only required for setting up and then later visualizing the experiment. Another closed-loop experiment was recently implemented by Corbinian Schreiber. A corresponding publication is in preparation. It discusses a path integration network, here shown on the right-hand side, which is inspired by respective circuits in the nervous system of insects. The network receives a simulated optical flow as well as the orientation of the insect. In this network graph, these inputs are shown in yellow. The populations below integrate these during the flight of the insect, and based on this integrator state, the insect is then autonomously steered towards its nest, or rather the starting position. The experiment starts with a fly-out phase where the tank drive-like motor units are controlled externally to perform a random walk, and after 60 milliseconds, which, taking the acceleration of the system into account, corresponds to 60 seconds of biological real time, the insect is released and returns home on its own. On the right-hand side, we show a trajectory from an experiment recorded from Brainscales 2. The path of the insect as well as the network activity are of course slowed down for visualization purposes here. But since the environment, including the sensor inputs, is simulated on the embedded plasticity processor, we can reach quite high repetition rates. So here you can now observe the trajectories from multiple successive experiments, which are now rendered in the actual walkbook time. In all of these experiments, the insect successfully returns to the starting position, and as soon as it has reached the home position, it circles around it. I think it's nice to note how the gaps in between the individual runs correspond to the time required to read out the data from the ASIC. The fourth experiment, designed by Benjamin Kramer and myself, attempted to address the issue of limited fan-in, which is almost ubiquitous in most, maybe even all neuromorphic systems. It certainly is present in analog systems where a more or less dedicated set of physical resources, specifically synapses, is allocated for each neuron. In some digital systems, time multiplexing can be employed to process a larger fan-in. This, however, often comes at the cost of increased simulation times, so you end up with a trade-off. To solve this problem, we can once again take inspiration from the nervous system, where a constant change of the connectome can be observed. Synapses do not only change in their strength, but some are removed and others are created continuously. In this experiment, we used such structural reconfiguration to learn a sparse connectome. We demonstrated this for a simple feed-forward network with a potential all-to-all connectivity between a set of virtual receptors and label neurons. This can be seen in panel A. By potential, we mean that each of the label units can connect to any of the presynaptic partners, but only to a subset of them at each point in time. In contrast, we call this subset the realized connectivity. We trained this network to classify the iris dataset. For this purpose, we reduced the data to only a two-dimensional feature space, which you can see in panel B. On this plane, we then randomly placed receptors. The connections were learned through Hebbian potentiation and a regularization term which fostered competition between synapses. This you can see in panel C, where the weights of exemplary synapses are plotted against time. The discontinuities show that weaker connections are regularly pruned, and in their place new connections were formed. To implement this structural reconfiguration, we exploited the fact that the synapse array of the BrainScales 2 system, very similar to BrainScales 1, was from the beginning designed for sparse networks. Each synapse does not only keep a 6-bit weight, but also a 6-bit label. This allows us to inject up to 64 different spike trains on a single row of synapses, and each synapse then attaches to one of these presynaptic sources. Changing the stored label effectively removes the previous synapse and instantiates a new one in its place. We implemented this rule on the embedded plasticity processor, and in our experiments it led to the formation of receptor fields which clustered around the data points. In the end, we were able to dramatically reduce the number of physical synapses without a measurable loss in the classification performance. The last experiment, performed by Julian Gölz and Laura Kriener, demonstrated the benefits of a time-to-first spike coding scheme for multi-layer networks of LIF neurons. To encode the inputs, in this case from a simple vision dataset, active pixels corresponding to higher values were converted to early and inactive ones to later spike times. The output of the network was decoded correspondingly. The first neuron to fire determined the classification response of the network. Julian Gölz derived a set of closed-form equations for the backpropagation of errors in such networks of LIF neurons, and he used these equations to implement an in-the-loop training algorithm which was based only on the spike times which we recorded from BrainScales 2. You can see a corresponding raster plot in panel C which shows the spike times of the whole network. The gray markers denote the hidden units, and the 4 label neurons are highlighted in color. And I think it's nice to observe how the first label unit fires long before the majority of the hidden neurons has emitted a spike. Panel D shows how the spike times evolved during training. The correct label unit is continuously pushed to fire at earlier times, and the other units fire at later times. Panel E shows traces from these exact same label neurons after training. They were recorded from BrainScales 2 via an on-chip ADC. These traces also highlight the low classification latency, which is in the range of 5 to 10 microseconds depending on the network parameters. We were able to train such a network on a 16 by 16 pixel version of the MNIST dataset, where an overall test accuracy of approximately 96% could be reached. More impressively, we measured an energy of only 25 microjoule per presented image. And here, please keep in mind that this substrate is very generic and by no means optimized for this task. To summarize, I think this set of experiments has nicely shown how BrainScales 2 can be employed to perform different kinds of studies based on the emulation of a wide range of network topologies. Importantly, our embedded processors can be programmed to efficiently implement various plasticity rules. All of the shown experiments have definitely profited from the accelerated nature of the system. In particular, this is of interest for the emulation of otherwise long-lasting processes. Structural plasticity, for example, in biology typically takes place on timescales of hours. On BrainScales 2, these can be rendered to only seconds in emulation. Despite this acceleration, we are still able to implement closed-loop experiments and even further tighten that loop to be fully contained on the Neuromorphic ASIC. In case you have any questions, or if you are interested in bringing your ideas or experiments to BrainScales 2, please feel free to contact us. I want to conclude by thanking all my co-authors and all other members of the Electronic Visions group in Heidelberg, and of course, most importantly, you for listening. Thank you very much.